Okay, so here we are at the start of the nervous system. Oh, 1 second, guys. This says part 2. Let me just make sure we're on the right. Here we go. I see what's going on. Okay. Thanks for your patience, guys.
Poor little guy got the hiccups.
Oh, sorry. What did you say?
I said your poor little guy's got the hiccups.
I know. Do you guys hear that? He's right by the microphone. So if it's distracting, let me know.
No, it's fine.
Okay. Thanks, guys.
You're welcome.
Okay. You guys are so nice. Thanks. Okay. All right, so here we are in part 1. We don't want to go to part 2 yet. That might be a little confusing. So part 1 of the nervous system. So your nervous system, simply put, we divide it into the peripheral nervous system, that's the PNS, and the central nervous system is the CNS. Your central nervous system consists of the brain and the spinal cord. And the peripheral nervous system will consist of all of the nerves that come off of the brain and spinal cord. So those will be the cranial nerves, the peripheral nerves, as well as the ganglia. And when it talks about ganglia, that refers to clusters of cell bodies. So that's what ganglia are. The nervous system functions. Your nervous system receives sensory input in all of the sensory neurons. It will integrate that information that it's perceived, and it'll integrate the information in your brain or your spinal cord. And then it will send out some sort of response to control your muscles and glands. So by doing this, your nervous system will maintain homeostasis and also will establish and maintain all of your mental activity, especially in the brain. So here are the divisions. Your central nervous system is your brain and spinal cord. The peripheral nervous system are all of the nervous tissue, nerves outside of your brain and spinal cord, like your cranial nerves and your peripheral nerves. We also divide the nervous system into what we call the sensory component and the motor component. The sensory division will conduct action potentials from sensory receptors to the CNS. So the sensory division is any sort of stimuli that affects your senses or anything in the internal environment. So that's what we mean when we say sensory division. And then the motor division will conduct action potentials away from your central nervous to all the effector organs. And those can be glands, they can be skeletal muscles. So simply put, you have a sensory division and a motor division of the nervous system. The part that senses stimuli, and then the part that performs some sort of response to those stimuli. The somatic nervous system, so we further divide our nervous system. The somatic nervous system will transmit action potentials from the central nervous system to your skeletal muscles. So the somatic nervous system is a motor division, and it's under conscious or voluntary control. Then the autonomic nervous system will transmit action potentials from your central nervous system to cardiac muscle, smooth muscle, and glands. And the autonomic nervous system is also known as the automatic nervous system. So these things will happen without you knowing it on the subconscious level. All right, and then the enteric nervous system is a special nervous system found only in the digestive tract. So here is kind of what we just talked about, but a chart that puts it into like a visual format for you guys to take a look at. Again, dividing the central nervous system from the peripheral nervous system and showing all examples of sensory input that will be taken up by the sensory receptors in your sensory division. So sensory input can include light. Light will touch your photoreceptors in your eye. Sound, taste, smell, touch, temperature, pain, pressure, any of the senses will be picked up by those sensory receptors and send it off into the sensory division, sensory neurons to your brain and spinal cord. The brain and spinal cord then will integrate that sensory stimuli, figure out what it is, process it, and then send out a response through the motor division of your motor system if it needs to. We divide the motor division into the somatic nervous system, which is under voluntary control, That means you guys can consciously control it. So the effectors will be skeletal muscle. So you guys control your skeletal muscles. You tell them where to move and when to move. The other division of your motor division is the autonomic nervous system. This is under involuntary or automatic control. And we divide the autonomic nervous system into a sympathetic or a parasympathetic division. Sympathetic is your fight or flight division. So that gets the body ready for emergencies. And your parasympathetic division is the rest and digest division. So its effectors will include anything internally. So again, things that are under subconscious control. This will happen involuntary. You don't tell your cardiac muscle when to beat. You don't tell your smooth muscle when to move things along your digestive tract. Those will happen without you knowing, which is a good thing. So all of these are described as the motor output, movement of your skeletal muscles, changes in metabolism, heart rate, breathing rate, etc. The cells of the nervous system, there's just two main groups of cells. They'll be either neurons and glial cells. The neurons are the nervous system cells that are excitable, that can carry an electrical signal through them. So your neurons will receive stimuli, conduct action potentials, and transmit signals to other neurons or effector organs. The glial cells, real simply put, these guys support your neurons. So you'll have different supportive cells in your brain, your spinal cord, and outside of that, in the PNS that just support the neurons. They do not conduct action potentials, so that means they cannot conduct the electrical signal that's being passed through them. Instead, these glial cells carry out different functions that enhance neuron function and also maintain normal conditions within nervous tissue. So I have a question for you guys. What is a glioma? Does anybody know what a glioma is? Or any guesses? Whenever you see the word oma, that usually means tumor. And glioma, that's a tumor of the glial cells.
It's a tumor on like the brain or spinal cord.
Yes, so if you hear of someone with a glioma, that means they have a brain tumor, and that's a tumor within their glial cells. So most often when people have brain tumors, it's always a tumor of your glial cells, never of the neurons. Good. So here is basic anatomy of a neuron. There's a cell body, dendrites, and axon. The dendrites are extensions from the cell body, and your dendrites usually receive information coming from another neuron and will transmit that information to the cell body. And the cell body will take that information received, process it, and then send it off down the axon. The axon is usually a single long cell process that leaves the cell body at the axon hillock, which is where the axon connects to the cell body and conducts sensory signals to the central nervous system and motor signals away from the central nervous system. So here's a look, some anatomy of a typical neuron. You can see the dendrites are extensions coming off of the cell body. There's your soma or cell body with its nucleus. Here's the axon hillock, which is the area that connects the cell body to the axon. And you might have collateral axons. Those are just branches off of the main axon branch. The axon itself will branch and end at the end. And those branches at the end will just serve to send off the signal to other neurons that will receive it. So that's a typical neuron. There's different structural types of neurons, and they all have to do with how many processes come off of the cell body. So a multipolar neuron has many dendrites and a single axon. This is one that is the most numerous within the central nervous system. Nearly all of your motor neurons are multipolar, and we'll focus a lot on the multipolar neurons. Bipolar neurons have two processes, one dendrite and one axon. Bipolar neurons will be located in some sensory organs, like the back of your eye and the retina, and also in your nasal cavity. The unipolar neurons have a single process that extend from the cell body. This will divide into two processes as a short distance from the cell body itself. One process extends to the periphery and the other extends to your central nervous system, and the two extensions function as a single axon. And here's a picture. So here's a multipolar neuron, the picture we initially showed to describe some of the anatomy of the neuron itself. And then a bipolar neuron you can see has a dendrite and an axon, and this pseudo-unipolar neuron appears to have an axon and no dendrites. And it kind of is shaped in the shape of a T with the cell body off to the side. So those are the types of your neurons, the structural component. So then we get to glial cells. And again, these are the supportive cells of your central nervous system. Astrocytes are the most common, and these serve as the major supporting cells. So astrocytes are the most common type of glial cell. They help to form what we call the blood-brain barrier. And that's because astrocytes have little tiny feet that wrap around capillaries. I think, yeah, we'll show you a picture of it in a second. But they help form the blood-brain barrier, and I'll explain why when we see a picture. The ependymal cells will line the cavities in the brain that contain cerebrospinal fluid. And cerebrospinal fluid is fluid that travels within the ventricles of the brain, but also travels down the central canal of your spinal cord. So they're all connected and they're filled with this cerebrospinal fluid. Microglial cells then act in an immune function in your central nervous system by removing bacteria and cellular debris. So microglial cells are kind of your garbage collectors. They'll remove all bacteria and waste products of cells. If a cell were to die, the microglial cells get rid of it. Oligodendrocytes are only in your central nervous system, and they provide myelin to neurons in the central nervous system. And myelin is important for kind of electrically insulating the neurons. It'll wrap around the neurons, the axons of the neurons, to help increase the speed of electrical conduction. The Schwann cells provide myelin to neurons in the peripheral nervous system. So Schwann cells do the exact same thing that oligodendrocytes do, but oligodendrocytes are only in the central nervous system and Schwann cells are only in the peripheral nervous system. And they both produce myelin to wrap around the neuron axons to help with the conduction of the electrical signal. So we say that axons that are myelinated will pass a faster electrical signal than non-myelinated axons. So these are the type of glial cells. Here are the astrocytes, and you can see they kind of have these big feet to them. They're called perivascular feet, and they'll wrap around capillaries. And when they do that, they form this barrier between what's inside the capillary, usually blood, but other potentially harmful substances traveling in your bloodstream. And they protect that from the tissue of the brain. So that's what the blood-brain barrier is. It's a barrier that astrocytes create that exists between the blood inside the capillaries and the brain tissue. There are some things that can pass through this blood-brain barrier. Nicotine, effects from alcohol, and that's how smokers and other drugs can pass through this blood-brain barrier. So this is why smoking, alcohol affect your brain so quickly, because it gets through this barrier of the astrocytes. Here are the ependymal cells. They have cilia on them. They will line the ventricles of the brain that contain the cerebrospinal fluid, so the cilia just helps keep it moving. Here's a microglial cell. Again, that's like the garbage collector. It'll engulf any bacteria that needs to get taken out of the brain. And here are oligodendrocytes wrapping around axons. They will produce the myelin sheath in your central nervous system. And then here are Schwann cells in the peripheral nervous system. And then what you're seeing here are layers of this myelin sheath that both of these cells produce. And again, think of myelin as just an electrical tape insulator that helps pass on the electrical signals faster. All right, any questions so far? Okay, I just muted everybody. I heard some background. Okay, So here's a little more detail about the myelin sheath. These are specialized layers that wrap around the axons of some neurons. Those neurons that have myelin sheaths around them, we say they are myelinated. And the sheaths are formed by oligodendrocyte cells in your CNS and Schwann cells in the PNS. Myelin produces this excellent insulation that prevents almost all ion movement across the cell membrane. And that's important because with no ion movement, that means the electrical signal will pass more quickly. Gaps in the myelin sheath are called nodes of Ranvier. So the myelin sheaths don't wrap around the entire axon. There's gaps or spaces between them. They occur about every millimeter, and the ion movement can occur at the nodes of Ranvier. All right, myelination of an axon increases the speed and efficiency of the axon potential generation along the axon. And multiple sclerosis is a disease of the myelin sheath that causes loss of muscle function. Are you guys still there? I just lost you.
Yes, I'm.
Okay.
Yes.
We're here.
Perfect. Thanks,
Yes.
I'm here.
guys. Thanks. I just
here.
lost seeing you all in my right-hand screen. So good. I haven't lost you. All right. So multiple sclerosis, it's a disease. It affects most often women and younger women. And what it does is it produces these structures called scleroses that perform a hardened, kind of hardened plaque along your axons. And that that basically decreases the electrical activity that passes through. Okay, unmyelinated neurons lack the myelin sheaths, and these axons rest in indentations of the oligodendrocytes in your CNS and the Schwann cells in your PNS. A typical small nerve, which consists of axons of multiple neurons, usually contains more unmyelinated axons than myelinated ones. So here's just a look at a myelinated and an unmyelinated axon. You can see how the myelination wraps completely around the axon versus an unmyelinated axon will have spaces or gaps between the myelin. Okay, then this is a look at how we organize nervous tissue. The nervous tissue varies in color due to abundance or absence of myelinated axons. And we call nervous tissue either gray matter or white matter. And really simply, white matter contains axons with myelin sheaths because myelin is white. So white matter will contain those myelinated axons. And gray matter will consist usually of neuron cell bodies and their dendrites where there's very little myelination. So that's gray matter and white matter. Then we'll talk a little bit about membrane potentials. Yeah.
Is that only going to be found in the brain?
Gray matter and white matter?
Yes.
Good question. There's gray matter and white matter only in the central nervous system. Good. Yeah. So the gray matter and white matter will be in your brain and your spinal cord, but all of your peripheral nerves, your cranial nerves that come off of the brain and spinal cord, they are just bundles of axons that are leaving that. But yeah, gray matter and white matter is only in the brain and spinal cord.
Thank you.
Good question. Okay. Resting membrane potentials and action potentials occur in neurons. Again, an action potential refers to when a nerve fires off an electrical signal, so it's been electrically stimulated. And we talk about a membrane potential refers to the electrical charge that exists on the inside of an axon. So on the inside of an axon at rest, it's about -70 millivolts. So that's the electrical charge of it. These potentials are due to differences in concentrations of ions across the membrane channels, and the sodium-potassium pump. So a resting axon tries to keep the inside of it at -70 millivolts. That's the electrical charge at resting. And it does that through the use of ions, membrane channels, and sodium-potassium pumps. The membrane channels include leak channels and gated channels. Leak channels are always open to ions passing through them, And gated channels are generally closed and they can only be opened due to voltage or chemicals. And that they'll usually only open when the neuron is being electrically stimulated. So a leak channel, these will always be open. And again, we're talking about membrane channels that are on the membrane of an axon. And we're getting to the point where we'll talk about how that axon gets electrically stimulated to fire an action potential down it. Because there are 50 to 100 times more potassium leak channels than sodium leak channels, the resting membrane has much greater permeability. That means potassium channels will leak through much more easily than sodium. That's Na. Therefore, your potassium leak channels have the greatest contribution to the resting membrane potential because they're the ones that are leaking through more easily. Gated membrane channels are closed until they are opened by specific signals. Chemically gated channels are opened by neurotransmitters or other chemicals, whereas voltage gated channels are opened by a change in membrane potential. And that change in membrane potential occurs when a nerve gets stimulated. So by some sort of external or internal stimulus that is stimulating that neuron. When these channels are opened, the gated channels can change the membrane potential when ions rush through them and are thus responsible for an action potential. And again, an action potential just means that neuron has fired or it's been electrically stimulated. Then we get to the sodium-potassium pump. And the sodium-potassium pump is a transport mechanism that requires ATP, so it's an active transport mechanism. The sodium-potassium pump acts to help compensate for any sort of leakage of ions through their leak channels. So the sodium-potassium pump tries to keep the resting membrane potential at -70. And that's important so that if that neuron gets stimulated, it'll be more easily able to perform an action potential by these concentration gradients that are established with the sodium-potassium pump. So what exactly does it do? It helps to maintain a greater concentration of sodium outside the cell membrane, and it tries to keep potassium ions inside the cell membrane. So it actively transports 2 potassium into the cell and 3 sodium out of the cell. And that 2-3 ratio is just the ratio of the sodium and potassium pump transporting 2 potassium in and 3 sodium out. And that happens at the exact same time. It's estimated that the sodium-potassium pump consumes about 25% of all the ATP in a typical cell and 70% of the ATP in a neuron. And that's because in order for the sodium-potassium pump to function, it requires ATP to do that transfer of 2 potassium in and 3 sodium out. How are we doing so far? Questions? We're kind of talking a lot about the anatomy of the membrane, and then we'll tie it all together and talk about how an electrical signal passes through it. Okay, so the resting membrane potential exists because the concentration of potassium will always be higher inside the cell, and the concentration of sodium is always higher outside of the cell. The presence of other negatively charged molecules like proteins will remain inside of the cell. They're too large to exit the cell. And that's why the inside of the cell is always at -70, because a lot of these protons are negatively charged and they'll always remain inside the cell. And again, it's important just to remember that the presence of leak protein channels in the membrane will be more permeable to potassium than to sodium. So sodium will always diffuse into the cell and potassium will diffuse out. And that's because sodium and potassium will follow their electrochemical gradients. Because there's more sodium outside of the cell to begin with, when sodium channels open, sodium will just diffuse into the cell. And because there's more potassium at the resting point inside the cell, when potassium channels open, potassium will move out of the cell. And again, in order to maintain the resting membrane potential, the sodium-potassium pump will recreate this sodium-potassium ion gradient by pumping sodium out of the cell and potassium into the cell. So here's a look at the resting membrane potential. These are channels. This is kind of similar to what we did, or similar pictures to what we kind of went over with muscles on Tuesday. And this is showing, whoops, go back. This is at resting, all the channels are closed except a few leak channels. And you can see how there's a greater concentration of potassium inside of the cell. Those are purple, and there's a greater concentration of sodium outside of the cell. Those are pink. And these arrows, purple and pink, show the direction that potassium and sodium will travel when the channels open. Here is showing the leak channels and how there are more leak channels for potassium. That's in purple. So the purple potassium channels will leak out of the cell more frequently than sodium channels will leak in. And that's just because you have more potassium leak channels on the membrane. And again, this is a picture showing the membrane of an axon in a neuron. And these different channels are the leak channels that allow ions to leak through them. And axons have more potassium channels that are leaking potassium ions out. This shows the sodium-potassium pump that also helps to maintain resting membrane potential. And again, the resting membrane potential is the -70 membrane voltage. That's the electrical charge on the inside of the membrane. And you can see here, there's 3 little circles on this pump, and that refers to pumping 3 potassium out for every 2 sodium that it pumps into the cell. And here's an ATP molecule showing that ATP energy is required for that pump to function. Okay, so how does an action potential occur? An action potential will allow conductivity along the nerve or muscle membrane, and it's similar to electricity going through or along an electrical wire. So that's what an action potential is. The channels responsible for an action potential are voltage-gated sodium and potassium channels, which will be closed during rest. So when a neuron is not being electrically stimulated, these voltage-gated channels will be closed. But when a stimulus is applied to a nerve cell, and again, a stimulus could be pain, it could be heat, cold, touch, it could be a photoreceptor stimulus in your eye, that light is being projected on it. So any sort of stimulus, after that's applied to a nerve cell, following a neurotransmitter activation, the chemically gated channels will open. So the sodium channels will open very briefly, and sodium will diffuse into the cell quickly. This movement of sodium ions into the cell, we call that a local current, causes the inside of the cell membrane to become more positive. Because at rest, the inside of the cell membrane is very negative. If we add into these incoming positive charges, that makes the inside of the cell membrane become positive, and we call that change depolarization. If the depolarization is not strong enough, the sodium channels will close again, and the neuron will not conduct an action potential. But if depolarization is large enough, sodium will continue to enter the cell so that the local potential reaches a threshold value. And the threshold value will cause depolarization of the other voltage-gated sodium channels to open. And these generally happen at the axon hillock of the neuron. The opening of these channels causes a massive 600-fold increase in membrane permeability. So once we reach a threshold voltage due to a strong enough stimulus, it will cause like a positive feedback response, opening up all sodium channels. So sodium will rush into the cell, will reach an action potential, meaning the neuron has responded, it's fired to that stimulus, and voltage-gated potassium channels will begin to open. As depolarization continues at a much faster pace, causing a brief reversal of charge, the inside of the cell membrane becomes more positive relative to the outside. And this charge reversal will cause your sodium channels to close, and sodium will then stop entering the cell. So the action potential peak, we say, will occur when all sodium channels will close. Sodium will stop entering the cell, but during this time, more potassium channels will open and potassium will leave the cell, resulting in repolarization. I'm going to keep talking a little longer and then I'll ask questions and hopefully we'll get to this graph, which will explain the picture a little more. At the end of repolarization, the charge on the cell membrane will briefly become more negative than resting. And that's because during this period of time, the cell membrane has lost too many potassium positively charged ions. And we cause this condition hyperpolarization. It also occurs briefly. Action potentials occur in an all or none fashion. And that refers to the fact that if that threshold voltage is reached, meaning if a strong enough stimulus occurs, an action potential will occur no matter what. But if that threshold voltage is not reached, no action potential will occur. The sodium-potassium pump will also assist in restoring the resting membrane potential after repolarization occurs. So I'm going to explain what I just talked about in a graph-like format because we're able to measure the inside of an axon. So we can measure what happens to the inside of an axon, and this will hopefully help clear up some of the questions you might have, because I'm sure this is a little confusing. But here we are at resting membrane potential, -70. The neuron has not been stimulated. If there is a stimulus, it isn't strong enough to reach the neuron to threshold yet. And the threshold is at this dotted line. The threshold usually voltage is about -50 millivolts. And if the stimulus is strong enough to open up enough sodium channels where sodium enters into the cell, if we get past threshold, we generate a positive feedback response, which will open up all the sodium channels in that area. And sodium will rush into the cell, causing depolarization. Depolarization causes an action potential peak to occur no matter what. And that's because we've gotten the inside of the cell past this threshold voltage value. Once an action potential peak has occurred, all the sodium channels will close and potassium channels open. Potassium channels will open and potassium ions will leave the cell. And that period of time is called repolarization. When the potassium ions leave the cell, potassium channels remain open a little too long. And when we have all of these positively charged potassium ions leaving the cell, too many of them will leave and we get to a very negative membrane voltage. And it's almost too negative. It passes the resting membrane potential and we get into a hyperpolarization state. And the hyperpolarization state will be restored to resting membrane potential due to the help of the sodium-potassium pump that tries to reestablish the charge concentrations of those ions being passed back across the membrane. Okay. Questions? Are you completely lost? Type in the chat 1 to 10, 10 being you're feeling okay about it, 1 being you're completely lost. Type that into the chat and we'll go through these steps in detail one at a time now. Okay. I feel like the people who are lost are probably not writing it in the chat. So if you want to send me a private message, you can do that too with your number if you are lost. Okay. Keep them coming. I'll take a look at the chat as we're going through those. All right. So here we are, resting membrane potential. This refers to a period of time when that neuron is not being stimulated. So all of the channels are closed. Again, the pink channels are the sodium, purple channels are potassium. You'll see more sodium are concentrated outside the cell and more potassium are concentrated inside the cell. And at resting, the inside of the cell we call that resting membrane potential is at -70. So that's why you see -70 here on the graph. That's resting membrane potential. Depolarization will occur when sodium channels open and sodium, which is a positively charged ion, rushes into the cell. Since sodium is positive and it rushes into the cell, the inside of the cell becomes more positive, and we start to increase our positivity on this action potential graph. We have gotten enough sodium to bring the inside of the cell past threshold. So this is our all or none response. We will generate an action potential peak no matter what at this point. Repolarization occurs, and we see a downswing going back negative on the graph, and the inside of the cell becomes more negative because potassium channels are open, sodiums are closed at this point. Potassium channels are open, and the inside of the cell will lose all of our positively charged potassium ions. If the inside of the cell loses a lot of positively charged ions, the inside of the cell becomes more negative again, and it becomes almost too negative. So you can see here the potassium channels remain open too long, so too many potassium ions leave. So we get to a hyperpolarization state. We want to get back to a resting membrane state, and that's here where my cursor is. And in order to get back to that resting membrane potential, the sodium-potassium pump helps us do that. Okay, so we'll pass away from that kind of part for a while, but you should have a good understanding of what each kind of phase is in this graph, the 1, 2, and 3, what channels are open at each phase, and what ions are rushing into and out of the cell. So a little more about unmyelinated versus myelinated axons. Action potentials will be conducted more slowly in unmyelinated axons and more rapidly in myelinated axons. Action potentials along unmyelinated axons occur along the entire membrane, but action potentials on myelinated axons occur only in a jumping pattern at the nodes of Ranvier, and those are the spaces between myelin sheaths. This type of action potential conduction is called saltatory conduction because it'll jump between those nodes. So this is just a look at unmyelinated action potential. It'll occur down the entire length of the axon, and this shows how the charge difference changes as the action potential travels down the action potential. And again, we call that depolarization when there's a change in membrane potential. Versus myelinated action potential, these nodes of Ranvier, the action potential will jump node to node as the action potential is propagated down that axon. At these nodes, there's a higher concentration of sodium channels, and we've known from the beginning that the sodium channels need to open for depolarization to occur. So at these nodes, you'll have the concentration of sodium channels that will open for that saltatory conduction to pass the action potential down. Action or axon conduction speed. The speed of the action potential conduction varies widely, even among myelinated axons. So it'll be based on the diameter of the axon fibers. Simply put, axons with a larger diameter will conduct action potentials a lot more fast or faster than medium or small diameter axons. So regardless of whether they're myelinated or not, a larger diameter axon that is heavily myelinated will have the fastest conduction speed of about 15 to 120 meters per second. That's almost 300 miles per hour. So that's how fast your nerve impulses are traveling throughout the axons in your body. To put it in another kind of analogy, I think, what's the speed of planes, like 400 or 500 miles an hour? I'm not even sure. Maybe someone else knows. They can put that in the chat. Okay, so then we'll talk about synapses, and this is why this chapter is divided into 2 parts, because you can see there's a lot of information just in this one part. So we're almost done with part 1, and then I'll put a bunch more information in your brain when we get to part 2 on Tuesday. But a synapse is a junction where the axon of one neuron interacts with another neuron. That's what a neuroneuronal synapse is. We talked about a neuromuscular synapse where a neuron is connected to a muscle cell, but we're talking here about a synapse or a conjunction between one neuron to another. The end of the axon forms a presynaptic terminal, and the membrane of the next neuron forms the postsynaptic terminal, with a synaptic cleft or space between the two. Chemical substances called neurotransmitters will be stored in synaptic vesicles in the presynaptic terminal. When an action potential reaches the presynaptic terminal, or the end of that axon, that'll cause voltage-gated calcium channels to open and calcium moves into the cell. When calcium moves into the cell, this causes the release of neurotransmitters by exocytosis from the presynaptic terminal, and the neurotransmitters will diffuse across the synaptic cleft and bind to a specific receptor molecule on the postsynaptic membrane. Within the synapse, when those neurotransmitters bind to membrane receptors on the postsynaptic neuron membrane, this will cause chemically gated channels for either sodium, potassium, or chloride, depending on what type of neurotransmitter it is, to open or close in the postsynaptic membrane. It will cause them to open or close because different neurotransmitters can be excitatory, so they can cause that postsynaptic neuron to excite it to continue sending the signal down, or they can be inhibitory, so they can stop that signal where it is. The specific channel type and whether or not the channel opens or closes will depend on the type of neurotransmitter and the type of receptor. And like I said before, the response can either stimulate or inhibit the action potential in the postsynaptic cell. If sodium channels are open, the postsynaptic cell will depolarize and an action potential will result if that threshold amount is reached. If potassium or chloride channels are open, the inside of the postsynaptic cell tends to become more negative, and that will create a hyperpolarization in that postsynaptic cell. So that will usually inhibit the action potential from occurring. A lot of the most common known neurotransmitters are acetylcholine and norepinephrine. Acetylcholine is usually the neurotransmitter involved in neuromuscular junctions. Neurotransmitters do not normally remain in the synaptic cleft forever. So you won't have neurotransmitters just hanging out in the synaptic cleft, constantly stimulating a postsynaptic neuron. Their effects will be short-lived, and the substances in the neurotransmitters will be reduced in concentration when they'll be either rapidly broken down by different enzymes, or they'll be transported back into the presynaptic terminal where they came from. An important enzyme that breaks down acetylcholine is acetylcholinesterase esterase, and that will control the amount of acetylcholine that's in the synapse. And norepinephrine will be either actively transported back into the presynaptic terminal or broken down by enzymes. And these enzymes just help to control the amount of neurotransmitters that are in the synaptic cleft, just to provide greater control for how well your nervous system is passing along its signals. So this is finally a picture instead of a whole bunch of words. This shows the action potential traveling down the axon in the presynaptic terminal, so before the synapse. When the action potential travels past calcium channels, calcium rushes in, and the rushing in of calcium causes the synaptic vesicles with those neurotransmitters inside to release the neurotransmitters across the cleft. Those neurotransmitters being released across the cleft will bind to receptors on the postsynaptic membrane, and those postsynaptic membrane channels will then, once they bind those neurotransmitters, usually they cause sodium ions to rush into the cell and cause depolarization of the postsynaptic membrane. So what is a reflex? And I think this is the last thing we talk about. Pretty much we'll end with reflex, a little bit about pathways. A reflex is an involuntary reaction in response to a stimulus applied to the periphery and transmitted to the central nervous system. Reflexes will allow a person to react to stimuli more quickly than is possible if conscious thought is involved. Most reflexes will actually occur in the spinal cord or the brainstem rather than in the higher brain centers. And a reflex arc is the neuronal pathway by which a reflex occurs, and it has 5 basic components: a sensory receptor, a sensory neuron, interneurons, which are the mediators between the sensory and motor neurons. It'll include a motor neuron and then some sort of effector. And the simplest reflex arcs will not even involve motor interneurons. So, probably the best example of this, and again, a reflex arc is a way for your body to perform some sort of immediate motor response to some sort of stimuli. And I'm just going to give you an example. If you touch a hot stove, or if you touch something that produces immediate pain that your sensory receptors in your skin will receive, that will produce an immediate response and cause your effector organ, usually a muscle, to remove itself from that painful stimuli. So if you touch a hot stove, your skeletal muscle will immediately remove your hand from that hot stove. That sort of response is a reflex arc. It does not involve that sensory neuron sending its painful sensation to your brain. It just goes within the spinal cord and then sends out a motor response to your skeletal muscle. So these are the 5 parts of a reflex arc: the sensory receptor, the sensory neuron, the interneuron sometimes exists as the communicator between the two, and then the motor neuron performing its response on the effector organ. This talks a little bit about the neuronal pathway converging. The central nervous system has a simple to complex neuronal pathways. These are pathways between the neurons. A converging pathway is a simple pathway in which 2 or more neurons synapse with the same postsynaptic neuron. And this allows information to be transmitted in more than one neuronal pathway to converge in a single pathway. And the diverging pathway is a simple pathway in which an axon from one neuron will divide in synapse with more than one other postsynaptic neuron. And this will allow information to be transmitted in one neuronal pathway to diverge into 2 or more pathways. And we'll talk a little bit about this and why they're important, whether they will converge or diverge, and how that will help the transmission of a signal. So this is a look at converging. So you have 2 presynaptic axons converging onto one postsynaptic axon. And this is diverging, 2 presynaptic axons diverging its signal into 2 postsynaptic axons. Summation refers to a single presynaptic action potential. It usually does not cause a sufficiently large postsynaptic local potential to reach threshold. So many presynaptic action potentials are needed in a process called summation. And think of summation as like these action potentials piggybacking on top of each other to try to produce a stronger stimulus. The summation of signals in neuronal pathways allows integration of multiple subthreshold local potentials to occur, and the summation of the local potentials can bring the membrane potential to threshold and actually trigger an action potential. There is spatial summation that occurs when local potentials originate from different locations on the postsynaptic neuron, and there's temporal summation that occurs when these local potentials overlap in time. And this can occur from a single input that fires rapidly over time, which allows the resulting local potential to overlap briefly. Both spatial and temporal summation can lead to stimulation or inhibition. So they can both either stimulate that neuron to create an action potential, or inhibit it. So try to stop that action potential or signal from occurring. And that gets us through part 1 of this chapter.